Good afternoon, everyone, and welcome to the Rare Bone Disease Alliance Scientific Symposium, exploring fibrous dysplasia ossificans progressiva. I'm Melissa Bernardi. I'm the director of the Rare Bone Disease Alliance, and we are so glad to have you join us today. Just a few quick housekeeping notes before we get started. Please make sure to keep your your microphones on mute. And make sure to, if you have any questions, please feel free to put them in the chat and we will ask the questions during the Q&A sessions. We hope to have plenty of time for questions, so please bear with us. If we don't, we'll make sure to share our speakers' contact information at the end of the session, just in case you have some follow-up questions. Without further ado, I will go ahead and share and introduce Dr. Eric Rush, the chair of the RBDA Steering Committee.
Thank you, Melissa, and thank you for everybody who is joining us today. And I have the privilege of just getting to thank all of the people who really made this possible. And I'm here on behalf of the steering committee for the Rare Bone Disease Alliance, and so we're very excited to do a few of these events. They've always been of great interest and always been a lot of fun to put together. So I'd like to thank my fellow members of the steering committee and the rest of RBDA. I'd also like to thank the OI Foundation. Melissa's done a lot of work to put this on, and we really appreciate our partnership with her, our partnership with the OI Foundation, most definitely. We also appreciate our partnership with the FOP community, and we appreciate the sponsorship of Ibsen, who helps make this possible as well. So without further ado, I would like to turn it over to Michelle Davis, who is going to go over the agenda, and then we'll get started with our speakers. Thank you.
Okay, wonderful. Well, thank you so much, Dr. Rush. It's wonderful to be here today. The IFOPA and the FOP community really appreciates being a part of the Rare Bone Disease Alliance. And we are grateful for the opportunity through programs like the TeleEcho program, also sponsored by the Rare Bone Disease Alliance, to educate people about FOP and about the comorbidities and the ways that you can truly care for the patients that are living with FOP. Today, we're really excited to have the opportunity to share more about the research work that is being done in FOP. It's a really incredible research space. FOP is, Dr. Kaplan is going to tell you a little bit more about FOP if you're not familiar with it, but FOP is a 1 in a million disease, and there is incredible activity, research activity that's happened since the gene was discovered in 2006. We actually sit in a world today where the first drug for FOP was approved in the summer of 2023. Not approved everywhere, but approved in 3 countries. And there are currently 5 other active phase 2 and phase 3 trials. And that is due in great part to the work of some of the people that you are going to meet today, and their commitment to FOP and their commitment to researching ways to bring treatments to our patients. So today you are going to hear from Dr. Fred Kaplan from the University of Pennsylvania. He has spent his career serving FOP patients and researching this disease. He's going to be talking to you about the power of 1. And then following Dr. Kaplan, we will have Dr. Paul Yu, who is from the Massachusetts General Hospital and Harvard Medical School. And he is going to talk to you about a drug sitting on a shelf called saracatinib, and how it has gone from the bench to a phase 2 investigator-led trial, and the work that he and his team did to get it there. And then we're going to hear from Dr. Marlies Eekhoff, who is graciously joining us at 9 p.m. at night from the Amsterdam University Medical Center in the Netherlands. And she's going to talk to you a little bit about how she has used an innovative model to fund an investigator-led clinical trial. So we're excited to share with you about FOP. We look forward to your questions. At the end, you will see a slide with all of their contact information, and they are pleased to answer additional questions that you may have. If you have additional questions for the IFOPA, which is the leading patient organization for those living with FOP and supporting and catalyzing research, you can reach us at together@ifopa.org. And I just would like to thank one last time the sponsor of this program today, Ipsen Pharmaceuticals. So Dr. Kaplan?
Thank you. Okay, thank you very much. I'm going to speak to you today about about FOP, of course. And for those of you who don't know about FOP, I'll give you a little introduction. I have no financial conflicts of interest, and the title of my presentation is The Power of One When Something Expected Doesn't Happen. The center stage for FOP is childhood. This is the first child I saw out of nearly 1,000 patients over the past 40 years. And you can see the telltale malformations, the great toe, abnormal joint formation in the interphalangeal joint of the great toe, which is present in all classically affected individuals. And then about ages 2 to 5, painful lumps that look like tumors begin to appear. And then they mature into heterotopic bone, ribbons and sheets and plates of bone, replace the span the length of the axial and appendicular skeleton eventually, and make movement eventually impossible. This is a teenager with FOP, and you can see there the ribbons, sheets, and plates of bone that envelop the chest wall and make breathing very difficult. And on this CAT scan, you can see the ribbon, sheets, and plates of bone that involve not just skeletal muscle, but also a tendon, ligament, fascia, and aponeurosis. This is a disease that I said affects children, but this is the end result of FOP. This is Roger Zumfeld from Germany, who since passed away, but can't move any joints. FOP is really a metamorphosis, and from patients who've been misdiagnosed as having tumors, we can see in the early FOP histology, when we got these specimens, we can see incredible inflammatory cells, neutrophils, mast cells, macrophages that surround the vessels, invade the skeletal muscle, and eventually leave a wasteland of cells. There are stem cells in this tissue, fibrodysplasia progenitor cells, which we now know that explode into fibrous tissue, into cartilage, into bone, and endochondral bone formation is present in in all of the heterotopic bone that's formed in FOP patients. And as Michelle said, in 2006, my colleague Eileen Shore and I and Dr. Zasloff found the gene for FOP in our colleagues around the world. This is Jeannie Pieper, the president and founder of the FOP Association, and Stephanie Snow, whose family supported a lot of the original research. And we can see there the gene, the mutation that causes FOP. And we reported this in 2006, a recurrent mutation in the BMT type 1 receptor, ACVR1, causes inherited and sporadic FOP. We had 7 small families around the world, but most of the patients have a sporadic spontaneous mutation. This mutation leads to a heterozygous missense activating organ of function mutation. The protein is leaky. It's not constitutively active. It's only mildly constitutively active, but it's ligand dependent. And soon after the discovery, the New York Times printed this story, finally, a sense of exasperation, finally, with genetic discovery, hope for escape from a prison of bone. And the FOP gene is a powerful target for drug discovery. And as you'll hear from Paul Yu and Marilisa Eckhoff, the center of this bullseye here is really ACVR1 and inhibiting ACVR1 through kinase inhibitors. And immediately when we discovered the gene, we had the blueprint for the targets and the potential treatments. But if the gene was able to speak, it would say, I am the architect, not the builder, just like in a section of Moby Dick. I am the architect, not the builder. And as Avi Loeb, an astrophysicist from Harvard University, said, if you don't allow for surprises, you won't learn something new. And just as this man is looking through the telescope, there in the sky, his wife is pointing out, no, look here. There's something on the horizon that you need to see. And of all the 1,000 patients, almost 1,000 patients I've seen with FOP, there was this remarkable history of a man with FOP. I saw him many years ago when he was 21 years of age. He was fine. He had a normal childhood. He had normal growth, normal development, immunizations that didn't cause any problems, intramuscular injuries, dental work, no problems at all, until he was 21 years of age when he had a viral illness, and soon thereafter had an episode of neck swelling. He went to his local doctor. They thought it was a lymph node. He sent him for a biopsy. The biopsy did not show any lymph tissue. It showed fibromatosis, and the pathologist was a very astute pathologist, said this could be an early lesion of FOP. The report went back to the doctor, the general practitioner, who sent him to genetics, and the geneticist looked at his toes, saw the malformed toes, and did the FOP gene test. He had the classic mutation, and the young man said to him, well, what should I do about it? And he said, go to Philadelphia. There's a guy there that will tell you what to do. So he came to Philadelphia, and I saw him with a medical student, and I saw him from across the waiting room, and he was walking. He didn't have any problems. And I said to the medical student, this man doesn't have FOP. I saw him again. When I saw him, I repeated the genetic test, and I confirmed that he had the mutation. And I spoke to him, and I said, you live with this for 21 years. Just be very careful. Do not have any biopsies. By the way, he did not form any bone from that biopsy in his neck. And if you have any problems, come to see me. He came to see me again when he was 26, and this is what he looked like.
Hi, I'm Andrew Davis. I'm 26 years old. I live in Birmingham, Alabama. I'm now going for a theater degree at the University of Alabama. I was recently diagnosed with FOP at the age of 21, and it really didn't affect me until then. Andrew, can you look up at the ceiling? And can you look down? And turn to the left and to the right. Good. Let's see you bring your arms up over your head. Can you do that? That's excellent. And let's see you extend your elbows and bend them up. Good. And your wrists back and down like this. Good. And let's see you may need to hold on to the wall here. And let's see you bend one hip up and extend it. Good. And let's see you bend the other hip up and extend that. That's excellent. And let's take a quick look at your thumbs.
You can see there that he is missing the interphalangeal joint.
Down? Good.
The thumbs, about 50% of FOP patients have that.
We may take a look at your toes. It's actually just one toe. Yeah. Very good. Okay, and can you bend that for me?
See, you're missing the interphalangeal joint.
Thanks very much. Yes, sir. No problem.
2 days after I saw him, I wrote in the chart, I saw a 26-year-old man last Thursday who has the classic FOP mutation and classic malformed great toes, but essentially normal range of motion. He has a few tiny nibbons of heterotopic bone, but almost nothing. He is one of the most interesting FOP patients I have ever seen. Clearly, the mutant gene was expressed during embryonic development because his toes and his thumbs are lacking joints, but something is protecting him postnatally without making him sick. If we could figure this out, we would have a substantial treatment. Here's the gift staring us in the face. Stephen Friend and Eric Schatt wrote in the journal Science, Clues from the Resilient. Understanding how individuals remain healthy, resilient to disease may provide insights into disease pathogenesis and new treatments. We described him as patient R. You saw his name was Andrew, but patient R had the classic developmental findings of FOP, but a paucity of postnatal HO. You can see there that he had the mutation, the classic mutation. You can see that he had very little heterotopic bone, and his CAGES score, cumulative analog joint involvement scale, the lower the number, the better the range of motion. He had a score of 2 out of 30, but his age-related median was 18 out of 30. He had a whole body CT scan, and the volume of the HO for him was 47,000 millimeters of heterotopic bone, but the mean for 13 subjects of his age was 434,000 millimeters. So he had about 10% of the bone you'd expect for someone his age, or 90% less. We thought that there was a unique uncoupling. We said that there was he had classic congenital malformations of the toes and the thumbs. He also had some fusion of the joints of the C-spine, not from heterotopic bone, but from orthotopic bone, which we see as abnormal joint development, much like the toes, and osteochondromas. So the mutant allele was active during embryogenesis, but he had little postnatal HO, and our hypothesis was that he had insufficient inflammatory trigger. We've known from many, many studies done by our group and many, many other groups that the FOP gene mutation doesn't turn a teddy bear into a raging grizzly bear. It turns a teddy bear into a sleeping grizzly bear, and something wakes that grizzly bear up. And what wakes it up is inflammation. And is it possible, we asked the question, is it possible to identify a factor from the study of patient R that would allow us to interrupt the inflammatory trigger of FOP without shutting down the entire immune system? Sort of like having an atom bomb sitting on your living room floor. You might have the plutonium in there, but if you don't have a fuse to light it, it just becomes a conversation piece. It's not going to explode. And so we did a targeted biomarker analysis of 113 plasma proteins, most of which were inflammatory proteins. And we conducted that in 38 FOP patients and 39 controls. And out of 113 biomarkers, the only one that was significantly different in patient R was MMP9, matrix metalloprotease 9. What is MMP9? MMP9 is an enzyme. It's a protein. It's secreted by inflammatory cells. It regulates inflammatory proteins, including IL-1 beta. It modulates multiple inflammatory pathways. It promotes inflammatory cell attraction, inflammatory cell infiltration, and inflammatory cell differentiation. And it, very importantly, degrades and remodels the extracellular matrix and liberates inflammatory proteins, including activin A, which is bound up in the matrix to perlecan. And MMP9 cleaves that bond. And MMP9 orchestrates the migration of tissue progenitor cells in a large spectrum of physiologic and pathologic processes. So a patient R, we studied this in patient R. Here are 38 patients with controls in FOP patients, and we see a range, and there's no difference in MMP9 levels in the serum from in FOP patients from controls. But this is a logarithmic graph. This is a logarithmic graph, and you see patient R here is extremely low. He has way lower levels than other patients with FOP. He not only had decreased levels of MMP9, but he has decreased enzymatic activity of MMP9 compared to other patients and controls. And we did that, we assessed that by gel enzymography. And you can see that compared to other FOP patients, patient R had about 1/3 the activity of MMP9. And the question was why? And the answer is that patient R is a compound heterozygote for exceptionally rare MMP9 variants. We found this out by sequencing his entire genome. And we found that he got one MMP9 allele from the mother and one from the father. It was a compound heterozygote. We didn't see this in 31 FOP controls. The mother's allele was right at the junction of the signal peptide and the propeptide. And a structural biologist, Jay Graffi, thought that maybe this decreased a little bit of the secretion of MMP9, but the D165N was in the catalytic domain, and he predicted that this caused extreme disruption in secretion of MMP9 and also in the enzymatic activity of MMP9. And he predicted that this would decrease the activity of MMP9. This is a three-dimensional diagram of the MMP9 protein. And this mutation is right in the middle of the catalytic domain, which is basically like degrading the scissors and their cutting edge. We introduced each allele into a monocyte cell line, differentiated them into macrophages, and then polarized them into M1 and M2 macrophages. And we see that the A20V variant from his mother decreased it by a little bit, but it wasn't significant. But the D165N allele from the father decreased MMP9 secretion in a major amount. We also looked at the activity of these two alleles, and we did this again by gelatin zymography. The mother's allele didn't decrease the activity of MMP9, but the father's decreased it by about a half or more. And at this point, we knew why patient R had why he had decreased secretion of MMP9 and why he had decreased activity of MMP9, but we had no idea if that caused any decrease in his heterotopic bone formation. So in order to assess that, we obtained MMP9 knockout mice from the Jackson laboratory. They have a mild skeletal dysplasia at birth, much like the two patients who have MMP9 deficiency that have been reported. They have knockout alleles of MMP9. They have a mild skeletal dysplasia called anadysplasia, which corrects in several months, and the mice correct in several weeks. And we bred those MMP9 mice, which are healthy and reproduce extremely well. We bred them to our FOP mice, so we had an FOP mouse colony that did not have MMP9. We see here on the right the MMP9 knockouts with no MMP9. The heterozygotes have about half of the amount of MMP9, and the MMP9, the FOP mice with a normal MMP9, you can see here. And then we induce heterotopic bone formation with an inflammatory injury, and the MMP9 knockouts amazingly formed no heterotopic bone, none. The MMP9 heterozygotes also formed no heterotopic bone. We didn't believe this, and we repeated this, and we found the same results. So you need MMP9 to form your normal skeleton, but you need a lot more MMP9 to form your second skeleton. We looked at this histologically, and genetic ablation of MMP9 promotes regeneration of skeletal muscle in injured FOP mice. This is FOP mice with MMP9, and we see bone formation and cartilage, the normal the endochondral heterotopic ossification. And there in the FOP mice without MMP9, we see central nuclei and regeneration of muscle after 2 weeks. We asked the question, we didn't believe these results, and we asked the question, are there any drugs that are currently available that decrease MMP9 without having a mutation in MMP9? And we did a Google search, and to our surprise, the tetracycline class of antibiotics inhibit MMP9 transcription, translation, secretion, and activation at submicrobial doses, antimicrobial doses, independently of their antibiotic properties. They confer, the tetracyclines confer resilience to diverse inflammatory conditions in which MMP9 has a pathogenic role, including aortic aneurysms, muscular dystrophy, multiple sclerosis, and amyotrophic lateral sclerosis, ALS. Tetracyclines improve muscle regeneration in muscular dystrophy, and as you'll soon see, diminish HO in an FOP mouse model. And so we treated our FOP mice with minocycline, a derivative of tetracycline, and again, very little, if any, heterotopic bone. And we used the QTOA7D model. This is a mutation that doesn't exist in humans, that was developed by, it's a constitutively active mutation. And we see that even without ligand, minocycline decreases MMP9 and decreases the heterotopic bone formation. Again, we didn't believe these results, and we asked a friend at a pharmaceutical company that made an antibody to MMP9, a very potent human antibody, if they could give us a mouse antibody that's less potent, unfortunately less potent, to see if there was proof of principle that a mouse antibody could decrease injury-induced heterotopic ossification in FOP mice. And again, we saw that the mouse antibody decreased the heterotopic bone formation. And again, we saw in a constitutively active model that does not exist in humans that, again, we got decreased bone formation. So we have MMP9 inhibition by genetic, pharmacologic, or biologic means in multiple mouse models, abrogates trauma-induced heterotopic ossification, and leads to regeneration of injured skeletal muscle. And the question is why? And so again, we did CRISPR-Cas9. We inserted the two alleles from patient R, the maternal allele for MMP9, the A20V mutation, and the D165N mutation into monocyte cell lines, differentiated them into macrophages, and then polarized them into inflammatory and anti-inflammatory macrophages. And we looked at activin A specifically because activin A has been shown to be one of the very potent inflammatory activators of the mutant allele, the mutant ACVR1 allele. It basically turns a brake pedal into an accelerator pedal, and we asked if either of these alleles decrease activin A, and very surprisingly, the A20V decreased it, but the D165N allele from the father almost decreased it to nil. And we also did this in mice. We asked, is activin A secreted in the mice? It is secreted, and it's deposited in the matrix, and it cannot be it cannot be released from the matrix in MMP9 Mo mice, and so it cannot activate the ACVR1 allele, the ACVR1 mutation. So the mechanisms, certainly a lot of work needs to be done in this. This is just a very brief look at this, but MMP9 deficiency inhibits heterotopic ossification by activin A dependent and by activin A independent mechanisms. Marshall Urist, who discovered the bone morphogenetic proteins, said there are 4 cardinal requirements for endochondral heterotopic ossification in any context. He said this in 1965. He said you need an inflammatory trigger. He said you need an inductive signal. He said you need a receptive progenitor cell, which you now know are the fibrodipogenic progenitor cells in the skeletal muscle. And he said you need a conducive microenvironment. And MMP9 likely functions in all 4 domains. We see that in this schematic, this hypothetical schematic. We have injury, inflammation, and inflammation induces neutrophils and macrophages. The macrophages secrete large amounts of MMP9. Patient R, genetic knockdown, pharmacologic inhibition, and biologic inhibition inhibits MMP9. and MMP9 acts on the extracellular matrix on type 1 and type 4 collagens and activin A, heparin sulfate proteoglycans that cleaves this bond, and other active molecules as well, VEGF, which is very important for the formation of endochondral ossification. It acts on activin A, and it probably acts, we didn't show this, but it probably acts on the fibroadipogenic cells in the matrix. Any one of in the environment of the inflammation, in the environment of the injured muscle, and any one of these factors can inhibition of any of these factors can lead to decreased HO. So a conducive microenvironment, an active signal, progenitor cells, and obviously inflammation. And so how does one build a second skeleton? So ACVR1 is really the architect of the second skeleton, but MMP9 is, I shouldn't say it's the contractor, it's a contractor, a general contractor. It probably recruits it has many substrates, and it probably recruits many different substrates which are necessary for endochondral bone formation. And so the conclusion of this is MMP9 links inflammation to heterotopic ossification. It orchestrates an existential role in the pathogenesis of FOP, and it illustrates that one patient's clinical phenotype can reveal critical molecular mechanisms of disease that unveil novel treatment strategies. And so MMP9 links inflammation to heterotopic ossification, and so it can be placed on this schema of targets and potential therapies of MMP9 inhibitors. It links inflammation to heterotopic ossification, and probably upstream and maybe downstream also in spontaneous heterotopic ossification as well. Recent studies showing heterotopic ossification in a non-FOP model also shows that MMP9 inhibition dramatically affects heterotopic ossification. So we published this paper earlier this year in the Journal of Bone and Mineral Research that matrix metalloprotease 9 deficiency confers resilience in Fibrodysplasia Ossificans Progressiva in one man, and in mice. And there was an editorial written about this by Mark Wien and Yingxi Yang from Harvard Medical School, that actual disease insights from bedside to bench, not bench to bedside, but bedside to bench, and hopefully back to bedside in FOP. So what's next in the clinic? Well, we anticipate a clinical trial later next year, early next year, actually, with a powerful monoclonal antibody against MMP9, human antibody that's been tested in other conditions as well. That's been shelved and probably and has gone through phase 1 clinical trials and phase 2 clinical trials, and will be used in an FOP clinical trial. And we hope, if we can raise funds, that a clinical trial with available and affordable drugs that inhibit MMP9, such as the tetracycline class of antibiotics, also metformin and others that decrease MMP9, may be used as well. And we take our lessons from this from Dr. Yu and Dr. Eckhoff, which we're going to talk about, a repurposed drug in a few minutes. So we have many clinical trials that Michelle talked about. When we discover the gene, we have many targets and many possible treatments. Again, the center of the bullseye for FOP is the STOP-HOP trial, which Dr. Yu and Dr. Eckhoff are going to talk about, the Falcon trial, which is sponsored by Ipsen, and the Progress trial, which is sponsored by Insight. And so all these 3 clinical trials are looking at kinase inhibitors. The Lumina and Optima clinical trial, which inhibits activin A, that's sponsored by Regeneron. The Kyoto rapamycin trial, which is taking place in Europe. The PIV1 trial looking at palovarotene, which is very much downstream. And the anecaliximab trial, which is an MMP9 inhibitor, which is going to start very soon. And so MMP9 deficiency, as I said, confers resilience in FOP. And you've already met a superhero of the resilient. That's Andrew Davis. This is Mother Lorna. This is my colleague, Mona El Makadam, and my colleague, Bob Pignolo from the Mayo Clinic. We profoundly thank patient R and his parents for their magnificent devotion and commitment to this work for the benefit of the FOP community worldwide. I want to thank Vitaly Lunev, who did all of the mouse work and the CRISPR-Cas9 studies that I reported today, and my colleague Bob Pignolo, with whom I've worked on this project for over 10 years. I want to thank the Martucci family. This is Joe Martucci and Ashley Martucci as FOP, and her Aunt Joanne, and the Siegel family and the Marichek family. These are Joe's two sisters, and they kindly and generously supported the funding for Cure fundraisers, which took place in New York, which raised the money which allowed us to do this trial, and also the Radiant Hope Foundation and the Kelly family, which supported this trial, and then two anonymous families, one from Australia and one from Caldwell, New Jersey. So this was funded by the FOP families. And I think the lesson is this, that a single life, or an N of one, a single life can make a difference and change the lives of many. The power of one patient to shed light on a condition. We certainly think that there may be other things that confer resilience, FOP, but this was the one patient out of nearly 1,000 that I've seen who is very, very different, and certainly shed light on a very, very important part of the FOP story. So this is a van that I saw at the Philadelphia Conference Center. It said resilience and recovery for all, and I couldn't resist taking a picture of this van. It's a little scratched up, as we all are in the FOP world, but resiliency and recovery for all. And that's what we hope for, both with the kinase inhibitors, with the upstream, downstream, side stream, and inhibitors of FOP. And I hope that in several years, we'll have many, many tools in our medicine cabinet that we can use to stop this dreadful disease and cause nothing more than an inconvenience for patients who have this dreadful condition. So thank you very much.
Thank you, Dr. Kaplan. If people have questions for Dr. Kaplan, we can put them in the chat, and then Michelle or myself will go through and ask them of you. Give folks a few moments to write, if anything. All right, well, I think we'll go ahead and get started with Dr. Yu. Let's see. All right, Dr. Yu, if you would like to go ahead and share your screen, we can go ahead and get started with your presentation. And anyone who has questions for Dr. Kaplan can feel free to post them in the chat, and then we'll ask them at the end of the session today.
So much, Melissa. Thanks, Michelle. Thank you to the organizers for allowing us to tell you about this story in FOP, and really appreciate the great introduction to this disease and all the research efforts dedicated to FOP by Dr. Kaplan, who has certainly been at the forefront of this condition and of these investigational efforts for for several decades. So this is the story of saracatinib from the bench to a phase 2 investigator-led trial. And I'm presenting this on behalf of our collaborators, Alex Bullock and Marilee Seekoff, who you'll hear from. But we have been able to build this effort based on this really important finding from Eileen Shore, Matthew Brown, and Fred Kaplan, Mike Zasloff, nearly 2 decades ago, that FOP is caused by gain-of-function mutations in this BMP receptor called ACVR1 or ALK2 is the name of the protein. And what we have learned in that time since is that these gain-of-function mutations confer enhanced sensitivity to their intentional ligands, BMP ligands generally, that are involved in embryonic patterning, in iron homeostasis, in regulating inflammation and wound healing in many tissues, but that they have had a less understood role in mediating the balance between activin and BMP signals. And it turns out that these gain-of-function mutations render the proteins more sensitive to BMP ligands in general by a small margin, but render the cells that have this receptor mutation profoundly sensitive to activin, such that activin, which is normally competing with BMP ligands, turns out to activate bone-forming signals that would not normally be specified by activin. But the affinity for activin and D ligands seems to be encoded in the receptor and is evolutionary conserved across many species and serves some regulatory role that we're still trying to understand. But that regulatory role gets transformed into a signaling role that then triggers inappropriate bone formation in the right context. And when we started working in this field, we found, much as Dr. Kaplan and other clinicians working in this field have observed for a long time, that inflammation is the link between gene expression and the phenotype. When we globally expressed a gain-of-function mutation that's much more constitutively active, meaning always active as opposed to hypersensitive, in mice in all tissues, we could see that it was expressed in immune cells and in muscle cells and in these interstitial cells that we now know are the progenitor cells for FOP. But nothing actually happened unless there was a simultaneous injury, and that could be from muscle trauma, or in this case, through the injection of a virus that caused a viral muscle inflammation or viral myositis. And you can see the infiltration of immune cells in the spaces between these damaged muscle cells during this flare of myositis. And it was only under those contexts that the soft tissues would then transform into bone in this mouse model. And this is the first of several mouse models that have been developed for this. It is not the most authentic in terms of modeling that activin hypersensitivity, but it is a great model for understanding inflammation and muscle injury as a necessary second hit for developing this disease phenotype. So we explored in the same paper this concept that maybe inhibitors of the enzyme activity of this type 1 receptor that would impede its downstream signaling. This is a molecule that was developed in our lab that is a high affinity inhibitor of the type 1 receptors. It actually could prevent the injury plus gene-mediated heterotopic ossification in this model. You could see this within a month after injury, that the drug-treated animals were relatively protected from the vehicle-treated animals. And it turns out that even at this early stage of the research, you could modulate the phenotype by providing immunosuppressive therapies, in this case, a very high dose of corticosteroids, similar to what we sometimes use for flares in patients with FOP, could also suppress the formation of FOP lesions in this model, or FOP-like lesions. This was probably transient and required doses that would actually impair the growth of the animals, unfortunately, whereas treating with a kinase inhibitor could be sustained as long as the kinase inhibitor was continued well beyond the wound healing process for this injury and this inflammation. So this was, a decade and a half ago, and it's really been a hope from our lab and other labs that we could use this principle to create specific therapies for FOP that could be tolerable and effective. So that led to a more than decade and a half collaboration with a very talented structural biologist and biochemist from Oxford. This is Alex Bullock. And in parallel with many other efforts from our lab and commercial efforts from pharmaceutical companies, he did a very clever screen of known inhibitors of different kinase receptors. ACVR1 or ALK2 is a serine threonine kinase. It's important for shuttling signals downstream of the ligands of that pathway, the BMP signaling pathway, to instruct tissues to form blood vessels, airways, ducts, and even all of our organs, and even to distinguish between left and right when we're developing from embryos. So instead of screening hundreds of thousands of compounds, as sometimes drug companies do, he chose a very selective approach and only looked at a couple hundred compounds that are known to be semi-selective inhibitors of these kinase receptors. They occupy the pockets that are responsible for cleaving ATP and phosphorylating downstream targets. So basically, they compete for the natural fuel for these kinase receptors and interfere with their ability to then activate the proteins that would normally fall downstream. And one small molecule from the several that he screened turned out to have some interesting properties. That was saracatinib. Saracatinib is this small molecule that had actually been developed for a lot of more common diseases than FOP. It had been developed for several cancers and had not great success in terms of improving outcomes in cancer, but it turned out to be a pretty useful compound. And in addition to its known effects in binding to members of the SRC, or what we call the SRC family kinase proteins, it also bound to our protein of interest for FOP, ALK2. And in fact, if you looked at the top 5 proteins that this molecule inhibited, sandwiched right in the middle of the different SRC or SRC family kinases was our protein of interest, ALK2. And so looking back at the history of this compound, if FOP had been on the list of goals for this chemical, this pharmaceutical development plan, it could have even been given a name specific for not SRC, but for ALK2 kinases. We used a more authentic model provided to us by our collaborators at Regeneron. This is an Enochian mouse model that actually expresses normal levels of the mutation that's found in 95% of people with FOP. This is the R206H gain-of-function mutation, which is active and sensitive. And we use this to develop an injury-based model where muscle injury and gene expression would result in bone formation within a month, sometimes less, in our control animals. And when we treated with different doses of this compound that Alex's lab discovered, saracatinib, we found that we could inhibit the formation of the bone, which you can see by these micro CTs compared to the vehicle controls. And you could also improve the range of motion. On the right here, this scale on the left shows the range of motion worsening if you treat them with just a control. And naturally, they progress towards the maximum score of 3, but they never quite get there during just 40 days of observation. But if you treated them with AZD at either 2.5, 5, or 10 milligrams per kilo, you could actually suppress the loss of function and also the appearance of bone by X-ray. And these are some of the fellows in the lab working with Alex and my group who contributed to this work. And we also wanted to know if these doses were fairly safe, not just in general for the mice, but these mice were induced to form bone early in life within 7 days of birth. And it turns out that a concern with the children who have this disease is whether or not a potent modulator bone formation might impact the formation of your regular skeleton. And so this experiment B was designed to show that these were the 3 doses in that experiment that I showed you. There was no impact on the growth of the mice by weight. And when we measured their femurs by their length or their bone mineral density, we found that it was unchanged even at the highest dose compared to our control mice. So this was reassuring. And in fact, maybe not surprising given the fact that this molecule had already been tested for safety in over 700 patients for different cancers. and at different doses, all the way up to 1,000 milligrams a day. That is quite a high dose. That was the maximum tolerated single dose. But in several diseases more recently, it had been tested at 100 to 125 milligrams once a day orally for two benign conditions, for Alzheimer's disease and lymphangiomatosis, a lung disease, a fibrotic lung disease. It turns out that it's probably not effective for those conditions, despite good reasons to believe so from preclinical models. And that happens sometimes. But in the clinical trials, the vast majority of patients who were given these doses were able to stay in the trial and tolerated it well. And so that led us to the STOP trial, where we're now testing this in adults to prevent heterotopic ossification. And Marilisa is going to tell you more about that shortly. But this is sort of a picture of the network involved in the STOP trial. Includes Alex, Marilisa, myself, Richard Keen in London, and a wonderful collaborator in Germany, Clemens Stockklausner, as well as two PhD students who have worked in the Amsterdam sort of trial center, Vincent Verhege and Bernard Smild, who have really been pivotal in advancing the program. So looking forward to Marilisa's comments on this. I just wanted to talk about another unmet need in FOP. We have obviously been successful in showing the potential efficacy when we're able to control the development of FOP by timing the treatment before injury. But this left unanswered what we can do for patients now who have grown to young adulthood and who developed a lot of immobility and reduced mobility, reduced function as a result of the FOP that forms in their joints, fascia, ligaments, and muscles, as Dr. Kaplan described. This is a young woman who was able to adapt to the permanent fixation of her ankle joint by using very large lifts in her shoes to provide stability. This is the MRI showing that several of her ligamentous connections in her foot bones have become solidly, have formed solid bone. And there's always a question of whether we can improve function. So we have several cases in the modern treatment experience where patients, for various compassionate reasons, have undergone surgery to remove bone as a result of FOP. This is a young woman who had fixation of one of her legs at the hip joint due to a bridging heterotopic ossification lesion bridging the upper part of our hip joint to the head of the femur. And with a lot of careful planning, surgeons were able to make a minimally invasive incision just to remove the bone and to minimize damage to the tissues and nerves and circulation. They were able to actually restore the ability of this woman to walk for several months. But unfortunately, as the long history of this disease had warned, this bone rapidly formed and actually formed a near perfect replica of the original bone, despite being almost completely excised. And after 18 months, had not only completely filled those spaces, but if we measured the volume, was actually twice the volume of the original bone and a very comparable density as it matured. This is not an uncommon experience. This was a patient treated in Marilisa's center who, for compassionate reasons, had heterotopic ossification removed from her jaw in order to permit eating a non-liquid diet, as well as to protect her airway. And this operation, also meticulously planned with minimally invasive techniques, was able to restore mobility to the jaw with intact nerves and circulation. But within several months, this bone reformed and not only replicated the original bone, but exceeded the volume of the original bone by some. Despite that, there was increased space between the incisors. This gave her the ability to eat solid food, to have better speech function, and to have better safety with her airway. And she was happy with the result, despite knowing the risks going in and having a less than permanent improvement in her function. This is another patient who was treated by our colleague in Mexico, who had bone removed from a knee joint and similarly good results for a month or two, and then rapid recurrence, and to the same degree, if not more than what started. This is a patient from Hyderabad who had a similar surgery as our first patient, with nearly complete removal of bridging hip heterotopic ossification. And within 6 months and even 2 years, the recurrence of bone was probably several 100% greater than the original bone. Okay, so what we know from the modern clinical experience is that surgical resection leads to rapid recurrence of bone. And despite some transient improvement and maybe even some permanent gains in terms of safety and quality of life, it's generally not recommended. It's not the answer for most patients. So we thought, could we model this, and could we see if one of our treatments might change the natural history? And so this is the natural history of what happens if we perform surgery on our mice. We can do some pretty careful microsurgical technique to make sure that the nerves and vessels are intact. And in fact, we can take a mouse that had dense heterotopic ossification and restore the ability of these mice to ambulate for about a month, after which the bone comes back, and it comes back and then some, more than the original volume. We tested our 3 doses that we used in our prevention trial for the saracatinib molecule. This is the AstraZeneca product molecule name. It's AZD530. And what we found was, in contrast to the prevention studies, the lower doses were less effective. But if we went to this top dose, we could prevent the recurrence of bone. We could document this by micro CT radiography. These are the orthopedic surgeons in our lab who perform these delicate surgeries. And like I showed you earlier, only the top dose was able to meaningfully prevent the formation of bone. Now, to model the real world scenarios where we give this drug, we gave the drug only for 3 months and let the mice continue ad lib after that without any treatment. And we were worried that this would rapidly regress to the controls, but it turns out that there is some margin of protection even after 90 days of treatment. We do want to go back and treat for longer and see if we can keep this suppression longer. But it does seem that despite things looking pretty calm on the surface, you might need to suppress for longer. And maybe if it's shown to be effective as a preventative, you might want to continue at a lower dose long term. This is the range of motion that was salvaged as a result of the treatment. And you can see the animals on vehicle would rapidly deteriorate after 1 month, but the animals treated with drug would retain the range of motion. People have asked, does this interfere with wound healing? And it turns out there's no meaningful impact on the closure of wounds after this surgery. This is in mice which are not great models of wound healing, but it turns out that this is very similar to the human experience with wound healing in the few instances where individuals in the cancer trials have had to undergo surgery or biopsies on this drug. So just in conclusion, saracatinib, it's an investigational drug that we're now repositioning because it's never been approved for preventing heterotopic ossification and FOP based on promising animal model data. The STOP-FOP trial is what Marlies is going to tell you about. We still keep thinking about how we can help those patients who have already lost some function, if there's an ability to restore it. And we are now exploring whether or not this treatment could be used to prevent the recurrence of FOP after surgery. I want to conclude just by acknowledging the generous support of many of our funders, including the NIH, IFOPA, the Department of Defense, the top row, second row, third row. These are all collaborators that have helped us in some way with modeling FOP or the biochemistry or the genetics. And I'd be happy to take your questions now.
Great. Thank you so much, Dr. Yu. We did have a couple questions get typed in for Dr. Kaplan, and I anticipate that there'll be some for you as well. It's just taking some people a little time to type. So I think what we should probably do is go ahead and move on to Dr. Eekhoff, and then we can do all of the questions at the end. Does that work for everybody? Great. And Dr. Ekoff, we're seeing your presenter view.
Now you hear me.
Oh, yes, but we're seeing your whole screen. So we're seeing the Zoom window now and the presenter view of your PowerPoint.
Do you see it now?
It's in presenter mode.
Yeah, so I can start.
Okay, so it's not the full screen mode.
I got the full screen mode, yes. As far as I can see, yes. Maybe this? Do you see it better now?
No, so it's showing us like your next slide. It's showing us the presenter view. So you have to, I think, switch it.
Okay, maybe Paul, maybe it's best you help me, you take over. Can you do that?
Yes, happy to.
As you discussed. Yeah, perfect. Thank you so much. Yes, thank you very much. Thank you for the invitation and being here and to talk about saracatinib trial to prevent FOP. Well, Paul just explained a lot, and I am the clinical part. We are performing this trial with a whole team. On the right side, you see here the team, Alex Bullock, and of course, Paul Yu, and on the left side, Richard King and Clemens Stocklausner. Next slide, please. FOP, or saracatinib trial to prevent FOP, is an investigator-initiated study. We got funded by the EU via the Innovative Medicines Initiative. We formed a consortium of several academic centers, as we just told you, Amsterdam, London, and Garmisch-Partenkirchen, together with the preclinical team members and with AstraZeneca. It is a combination of private-public. It means that the EU pays the money, but AstraZeneca is joined in kind, giving us the medication. Next slide, please. And of course, maybe you see it here also, the consortium works together with the stakeholders board. Very important, all the stakeholders are patients organizations, FOP patients, IFOPA, and also the Netherlands, United Kingdom, very important, Germany, France, Italy, Sweden, and even Spain. And they are also not only collaborating in the stakeholders board, but also supporting us on other ways and financially. Next slide, please. I'm not sure. Yeah. Well, this you just heard, just shortly, a very both of all the compounds Paul Yu and Alex tested. Saracatinib was by far the most potent. It was also shown that it is really good attacking the OCT2, the affected mutation, this X-ray crystallography. And you also see on the right side that the BMP signaling inhibition was shown with different dosages. And already Paul told, when you give FOP mice a vehicle, you see a lot of bone formation. But if you treat it with saracatinib in advance, you don't see or hardly see any bone formation. Next slide, please. Well, this very promising drug, saracatinib, was identified as a potential drug because it directly blocks region of the affected mutation. And because it blocks directly this mutation, it is potentially the most robust inhibition of FOP because it blocks the activin mu function, also the BMP hypersensitivity, but also blocks any leaking signaling. And we hope that if it blocks and if it works as we expect, it will be it will block not only both active flare-ups, but also the chronic heterotopic ossification formation. And if it works, as we hope, it might enable surgery in the long time, where you can unlock joints that has been locked because of all the heterotopic bone formation around it. Next slide, please. Well, here you see, we have showed it before, you can click several times, then you can see the activin and BMP can, I don't know if it will work, Paul, if you, yeah, it's going to work. It makes bone because it is the mutation, and where activin normally doesn't make bone, it does when it activates the mutation, the ACVR1 ALK2 mutated receptor. But, and then you can click several times again. Yeah, but if you give the drug, the kinase inhibitor, the activin A can't make any bone anymore. Next slide, please. As Paul told, and here is just a summary. The advantage of saracatinib is that it's a repurpose. Because it has been already investigated in so many healthy volunteers in cancer. It was originally developed for ovarian cancer. Then it was tested in other cancer, in Alzheimer and lung disease. And that's why we have over 700 patients tested and know that the drug is very safe. In STOP-FOP, we use the lowest drug as possible. And because of the data of Paul, that really shows that the engagement is enough for this. The aim of the study is to look, is a phase 2 study, and we'll show later, to look at the safety of saracatinib. We look at the side effects, blood results, but also we look into the effectiveness of saracatinib on FOP by using the 18-fluoride PET-CT, questionnaires, diary, and mobility. Mobility is a KG score. Next slide, please. The locations, 3 clinical sites, Amsterdam, Garmisch-Partenkirchen of Clemens Stock, Klaus in Germany, and Richard Keane in London. When we started in 2019, first of all, Brexit started, and then COVID. And that, well, that was a large hurdles to take, but we did. Only London couldn't manage, so now the study is only performed in Amsterdam and Garmisch-Partenkirchen. Next slide, please. The study is designed as a 6-month randomized controlled trial where you have 50% chance of getting the treatment with saracatinib and 15% placebo, and thereafter a 12-month open label extension phase where every patient has 100% chance to get the treatment with saracatinib. It gives us a possibility not only to compare the placebo arm with the drug arm in the RCT, but also to compare the placebo arm with the follow-up drug arm in the open label, and also to compare the open label phase with historical data. Next slide, please. And here you see PET-CT. The study, despite COVID, despite all the hurdles we had to take, the study is running very well. 17 patients are in Amsterdam in Germany. The study has been delayed for 2.5 and now 3 years. We have to change the slides. Of course, the inclusion is closed. The participants are doing very well. All participants who are in the study and already finished the study wanted to go on and wait for the results because they feel they benefit, but of course we don't know. The results will come next year. Next slide, please. We are very happy with the support and collaboration, not only with the EU IMI, but also, of course, with all the stakeholders, IFOPA, Spain, FOP, France, Italy, Netherlands, France, and others. Next slide, please. And here I give you a little bit background about funding and university-driven trial. Next slide. You can go on. Yes. If you look at the drug development, of course, it starts with a discovery. Saracatinib was discovered by AstraZeneca for another goal. And then preclinical research. And here we came in, Paul Yu and Alex Bullock, and found out that the drug was a very potent drug for FOP. And then we went to the clinical research, and I'll come by it later, where we didn't need to do the phase 1, but could immediately start with phase 3. And then the other 4 and 5, we are definitely not that far yet. Next slide, please. There's a beautiful picture of IFOPA. You can find it on their website, where you can see the phase 1. Well, we didn't need to do it, but if you start with a total new drug, you have to look in healthy people whether the medication is safe and doesn't have too much side effects. Then you go to phase 2, and there we are to test the drug in patients to see whether it's safe. Still very important. And you also look to the effectiveness, but the safety is most important. And when this works out, you go to the phase 3 to look at dosage and the final effect of the drug. Next slide, please. If you look at the difference between the industry-initiated clinical trials and academia-initiated clinical trials, then the academia, the doctor, needs to apply for a program or government grant to fund a trial. And of course, in industries financed by themselves. Next slide, please. In Europe, you can find funding in a country, but most national funding agencies doesn't accept funding across border zones or crossing borders. To change this, the European Committee created a European Research Area Network. There's a network where they try to get more, to combine the national sources and to support a coordination where different countries still can set up an international trial just by collaboration of the national funding agencies. But that is only started in 2020 and is still difficult. The Horizon Europe, of course, that is a very good funding program, but we went to the Innovative Health Initiative, the 4th one, because that gives you the opportunity to collaborate with industry and an already drug for repurpose. Next slide, please. Of course, if you look for more funding, you can go for charity organizations and patients organizations, as we do. And finally, you also look for collaboration with companies. Next slide, please. International funding. In Europe, only 3% of the academic trials involve more than one country, and it's because of the cross-border funding difficulties. The industry-sponsored trials are much more international, but rare diseases are so rare that as a consequence, you also always have to do international trials. Next slide, please. If you look at the different trial types, industries mostly initiate new drugs. Academia perform often comparative studies, but we did in between. We are conducting a repurposing study with a drug that is still under investigation, so a phase 2. And that is not that much often done. The execution industries use a CRO, a contract research organization, to perform the clinical trials, and they are responsible to find primary investigator sites. But as an academia, we did it ourselves. We are responsible totally for running the whole trial and to find other sites. It also means that we are very dedicated and put a lot of time in it. We need a lot of collaboration, but on the other hand, we have a lot of open discussion that makes it very wise and also very good to collaborate with the stakeholders. Motivations, well, both are the same, but it may differ a little bit from economical perspective. Next slide, please. Well, how do you set up a lot? And I think I can tell another time more about it. Protocol designs, plans, you make clinical teams so important, boards, very, very important, monitor, data manager, statistician, imaging team. We are all coordinating this. Safety recording, one of the mainstream in this trial, dissemination plan, future development, and a lot of work, but very important and good also for the trial. We wrote many reports for the financier. Next slide, please. Well, this last slide, and I took it from, as you can see below, on the website. It's very nice showing where we are looking for. We are looking for a class 1 drug. If you see on the x-axis and the y-axis, that you're looking for a selective drug, a high selective drug, so on the right side, and also a drug with a very high potency. And then you come at the class 1 on the right, and of course, all trials are looking for a class 1 trial with the lowest toxicity and the highest efficacy. Next slide, please. I think this is the last slide. Yes, well, that are members of our team of Amsterdam who are in one or another way involved and helping us a lot. Next slide. Okay, and this is my disclosure. Next slide, please. Questions? Yes. Thank you very much. It is late in the evening, but I don't know whether this is the reason that you don't see me that well in Amsterdam.
All right, thank you, Dr. Eekhoff, so much. So we do have several questions that have been posted into our chat. We can, I think, start at the beginning, which was a question for Dr. Kaplan from Matt Warman, asking, have you transplanted MMP9 KO hematopoietic stem cells into FOP mice to see if it is MMP9 from WBCs? That is essential.
It's a great question, Matt. We're doing that right now. Those are ongoing experiments. And obviously, MMP9 is made robustly by inflammatory cells, by neutrophils, by mast cells, by macrophages. And of course, those cells of the innate immune system are made by the hematopoietic stem cells. So we're doing a bone marrow transplant. We're doing bone marrow transplantation now, exactly like you said, from MMP9 knockouts into MMP9 replete FOP mice to answer the question, is it sufficient to knock out the MMP9 that's secreted by the inflammatory cells to decrease heterotopic ossification? Hopefully we'll get the answer to that question in several months. So that's a question we're doing. And if that is possible, it might be possible to take individuals' own bone marrow cells, knock down the MMP9 using CRISPR-Cas9 ex vivo, and give them back their own hematopoietic stem cells and treat FOP. So make them, in other words, a chimera. And you wouldn't need to immunosuppress them for the long term because you're giving them back their own stem cells. We got this idea from a patient many years ago who, interestingly, had FOP, and he had very severe flare-ups until age 7. At that time, the FOP flare-ups diminished and stopped, and at the same time, he was seen to be anemic by mother. He had increased infections and increased bruisability. He underwent a diagnostic workup and was found to have aplastic anemia, which we haven't seen in other FOP patients. He was given a bone marrow transplant by a sister, from his sister, who didn't have FOP, and she was a near match. He was immunosuppressed and cured his aplastic anemia. He was immunosuppressed for 15 years. His FOP was quiet, and unbeknownst to me, I didn't know him at the time, the prednisone, cyclosporin, and methotrexate were tapered and weaned after 15 years, and he was followed for 3 months. And then after 3 months, his FOP exploded everywhere, and he formed ossification. At that time, he had totally engrafted, and he was a complete chimera. His entire immune system was donor, and he had the FOP gene and all his connective tissue cells. And it proved to us, number one, that bone marrow transplantation doesn't cure FOP, that the cells that make FOP are probably fixed connective tissue cells, which we now know from amazing work by Paul Goldhamer, Paul Yu, David Goldhamer, Paul Yu, and others. But we also know that although FOP inflammatory cells may be more robust than other cells, you don't need FOP cells to trigger an FOP flare-up. And this patient taught us that even a normal immune system can trigger FOP heterotopic ossification in a genetically susceptible host.
Dr. Warman, any follow-up question to that, or everything?
Oh, you just didn't mention the other thing I said, which is great work and great talk. Nice to see you.
Thank you, Maggie. Very kind.
And for everybody in the FOP community, 25 years ago, all of this is completely unimaginable to most of us, but fortunately not to Fred and to the people who've persevered in bringing so many potential therapies into clinical trials. It's just incredible. I use it when I teach, and let's hope that we come to complete cures.
Thank you very much. And I think you heard from the other talks today that there's fabulous work being done, and hopefully, as I said, we can have a horrible nightmare disease, as Bridget Hogan said, and turn it into nothing more than an inconvenience. Maybe not in my lifetime, but hopefully soon.
Great. Well, thank you so much. So the next question is also for Dr. Kaplan from Vladimir Kennis. So thank you, Fred, so inspiring. I'm going to read the whole question this time. Do you think some of these medications, e.g. tetracycline derivatives, can work as a preventative measures in high-risk patients for neurogenic HO?
It's very possible. There's nothing about MMP9 inhibition that is isolated for FOP. So I think some of the treatments that we have for HO will be specific for FOP, some of the kinase inhibitors, some of the ALK2 inhibitors. But this one, probably not. And we now know our collaborators, other collaborators that we have tested this out in a burn tenotomy model. It's not neurogenic HO, but a burn tenotomy model and MMP9 inhibition, at least by genetic knockout, works in those animals. And so it's very possible. There have been a lot of great work done in the military. There's a lot of heterotopic ossification that's common, that can be caused by war wounds, by strokes, by head injuries, by hip replacements, by civilian athletic injuries as well. And interestingly, also about 13 to 15% of patients who have calcification of the aortic valve also have ossification in that valve. And about 13 to 15% of patients who have atherosclerosis also have bone formation, endochondral bone formation in their arteries as well. So it's possible that dramatically decreasing the inflammation can inhibit that. And as Paul said, steroids do that, but you can't use steroids, for long term. And obviously, a bone marrow, when you knock out the bone marrow, that does that as well. That stops FOP, and it may stop other forms of heterotopic ossification. But that's somewhat fatal. So it would be very, very helpful. That's what MMP9. I think that's the benefit of the MMP9 story, is that you can knock out a very specific part of the immune system that enables heterotopic ossification, because MMP9 speaks to many of these modulators and has many, many different substrates. So, who knows, but maybe, Vladdy.
Thank you. Dr. Yu, the next question was for you, from Brick Walker. He asked if there are pills available.
Well, we hope eventually there will be pills available. The phase 2 trial is designed to test the safety in these patients as well as the potential efficacy. And I think that in the next half year, we'll probably get our first installment of results from that trial. And I'm looking forward to that. Even though the subjective reports are always encouraging, you need to see the exact data. And so that the regulatory pathway that Marylise talked about is what lies beyond that. If you have a promising result, we may have to expand to phase 3. And that's usually a larger trial. And I think that's Matt's question. So if I might, Matt asks Marylise and Paul, exciting work. What is your primary outcome measure? Matt, maybe you wanted to ask?
Sure. Again, really exciting stuff. So I know that for the original trial of the inhibitor for the FDA approved drug, it was volume that was the primary outcome measure. Is that right?
So that is that's a retinoic acid gamma receptor agonist. Yeah, and that was the proposed endpoint. Yes.
And and again, who knows what FDA requires or what they will require going forward? But what are you using for your primary outcome measure? And how many patients do you think you're going to need to be powered to see it? Obviously, if you completely stop the progression of HO, then you need very few patients because you've completely stopped the progression. But if you moderate or ameliorate the progression without completely stopping it, how many patients do you think you're going to require?
Those are great points, and Marylise will give you some specifics, but you're absolutely right in that the FDA is aware that the state of the art is moving forward in trial design for FOP. And we've seen that in the evolution of trial designs from Clementia and then Ipsen with the approved agent palovarotene, which used this radiographic measure of bone volume by CT. And then with the Regeneron trial with garetosmab, they used that initially, and then as their trial evolved, they used possibly more reproducible measures that were related to the formation of bone, but actually to new bone lesions, new areas of involvement, as opposed to the actual, cubic millimeter volume of bone. But I'll let Marilise sort of answer what our approach is in the STOP-TOP trial.
Yeah, I think you can't see me very well here. Yes, as you said, Paul, I totally agree. The inside of the volume and the new lesions has changed. New lesions are very well, and if you look, and we use the PET-CT, the 18-fluoride sodium PET-CT, that's really of help to show the activity, the new activity, as well as the new lesions. If you look at speak about how many patients you need, well, it really depends. If the drug works very well, you don't need that much patients, and but well, we will know it next year. We look forward for the data. Yeah.
So you haven't done any intermediate data analyses yet?
No, we haven't. And that is the reason was the COVID and the Brexit, well, COVID mainly. So we haven't done that, no.
Because I asked because you'd mentioned the patients were doing well, but I assume by well you mean doing well in terms of no serious adverse events due to the drug itself. Yeah.
Got it. I do. So no serious side effects, and well, the patients say, well, we need to. The patients say they benefit, but we don't know, and we have to look at we have to look at the data. We have to wait for the data next year.
Great.
Yeah, and that's for all drugs, for all trials, of course. Yeah.
Great, thank you. And Marilise, this next question is also for you. This is, will studies of saracatinib be expanded to the US?
Yes, but maybe also Paul, he's part of the, Paul, you can answer.
Yeah.
We really want to do that, and there are a couple hurdles. One of them is funding for an American trial. And another one is licensing, so that whoever funds it might be able to obtain the rights from AstraZeneca. Those are being worked on. There are some groups that are interested in carrying it forward, and an American trial would be a logical next step, absolutely, and it would probably help with the phase 3.
Very good. Thank you. And next question is also for Marilise and Paul. This is another question from Matt. Thank you for the great questions, Matt. What is your primary outcome measure for your clinical trial?
I think we already talked about that.
Okay. And okay, so we'll go to the next question, which is also a question about saracatinib, and testing with adults or also with kids was the question.
Well, now we are testing first in adults. We always start in adults, and then of course the next step, but that totally depends on the next year. We want to go forward to study in children, yes, and to do, well, you have to do a lot of things before you can start, of course. Yeah.
Very good. Are there any other questions from the audience? All right. And Melissa, do you see any other in the chat, or did you get any other questions?
I've not got any other questions. Oh, someone just a comment.
Excellent.
Thank you, Laura.
Excellent discussion. Thank you for your collective work, and that's a great note to end on. And I would just like to thank our speakers, presenters for not only excellent presentations, but just also their hard work over the years in all of what they've done for FOP. I think Matt summed it up very nicely. And I haven't been doing bone disease as long as some people on the if you're on the chat, but I've been doing it for long enough that I've really seen just a great leap forward in what we're able to do and what we're able to look at with FOP. So it really gives our patients a lot of hope and a lot of promise. So thank you again to our presenters. Thank you to Melissa and the OIF for making this possible, and thank you to Michelle also for all of her advocacy for FOP over the years. And thank you to the audience for joining us and asking such great questions. And so with that, I will say thank you again to all and wrap up for the day and give you a little time back. And I hope everybody has a wonderful holiday season.
Thank you so much. Thank you, everyone.
Thank you.